Hey guys, welcome back. This is Lei. So far, we have covered most analysis in RNA-seq, ChIP-seq, and ATAC-seq. If you have followed along, you must have noticed that the hardest part of NGS analysis lies in the navigation of Linux system and the usage of command line tools. Unlike tools with beautiful graphical interface, command line tools are text-based. To most people, they are like black boxes. You don't know what's going on under the hood, what they are taking in, and what they are spitting out. The easiest way to solve this problem is to understand the data structure by directly visualizing the data. In this video, I'm going to walk you through the formats of common NGS data types, from raw sequencing data to alignment data to the quantification. Let's start from the raw sequencing format, the FASTQ format. They are the raw files we receive from the sequencing core or from the company. They are large text files and are usually compressed. Due to their large sizes, directly opening them on your computer will crash the entire system. But we can take a peek using the command less. In your terminal, type less your FASTQ file, and that will show you the inside of a FASTQ file. In the FASTQ file, each read is represented by 4 lines. First line is the header containing the IDs of the read and other meta information. We don't care. The second line is the actual sequence of the read. Third line is a separator line, usually repeating the first line or empty. The fourth line is the thread quality score line. The best for less is that you can also search specific sequence in a FASTQ file, for example, adapter sequence. So press / and type the sequence you want to search, and you can search and highlight the sequence. And this one is the adapter sequence I searched in my FASTQ file. So once you are done looking at the FASTQ file, you can press Q to quit. The second tool is called CIGAR. They can summarize the FASTQ file. So using CIGAR stats with your FASTQ file, the output is a TSV format file. So the top line is the column names. The bottom line is the information of your FASTQ file. Look at this. You have FASTQ, the DNA, type DNA, number of reads, sum of length, minimal length, 150, average length, and max length. Next, you can actually manually calculate the number of reads using cat command. That means I want to see the entire FASTQ files, everything in the FASTQ file. Here's a pipe. That means the output of the first command will be used as the input of the second command. The WC-L command means I will count how many lines in the FASTQ file. And divide by 4, that means I want to count how many reads are there in this FASTQ file. Pipe BC means calculating. So I will calculate this mass expression and output the results. And this is the number of reads in this FASTQ file. But if you use CIGAR, you don't have to calculate the read numbers by yourself. It already shows you number of seeks. So lastly, you can actually extract the first 4,000 entries in your FASTQ file and save it into a subset FASTQ file. Next is the FASTA format, which is used to store sequences, especially for genomes. Let's take a quick peek of our human genome file using less. Here is the content inside our FAST file. The name of the sequence is represented by the greater than sign and its name. Press Q to quit once you're done peeking. Of course, we have another command called more, but trust me, less is better than more, and is more than more. Just use less. Cat will show you everything in a file, but don't use cat for human genome file because it'll just print everything forever. We have SAM files and BAM files for alignment. SAM files are text files and are large, so we usually convert them into BAM files using SAM tools. The following sorting and indexing are required for subsequent analysis. SAM files can be directly picked in with the last command, but you can't do it for BAM files because they are binary compressed file. So we have to view them using SAMTools view command. But this command shows you everything in the file, so we pipe it to last. Only show a small portion of the BAM file. This is the inside of a SAM or BAM file. Lines started with S-line are header lines. They contain important information about sorting, reference genome, aligner, sample information, and the sequencing platform information. The last line contains the actual alignment information. Peeking into a BAM file is particularly important if you receive the BAM file from someone else and you want to know how the alignment was done and what tools was used or what samples was used for alignment. There is another popular alignment format, CRAM. It's the ultra-compressed version of BAM. It's much smaller than BAM files, it's lossless, and it stores only differences from the reference genome. That's why when you convert BAM files into CRAM file, you need the reference genome file. You can convert BAM to CRAM or CRAM to BAM using SAMTools. You can also pick into CRAM files using SAMTools, but you also need the genome file. You use CRAM file when you have limited storage. Now we have the expression matrices we are familiar with. They are either in TSV format or CSV format, the tab-separated format or comma-separated format. Inside of them, the columns are separated either by tabs or commas. If you directly look at them in terminal, they don't align well, but you can open them in Microsoft Excel and they will align perfectly. Lastly, we have BAD files, which are tab-separated files widely used in ChIP-seq or ATAC-seq data to represent peaks. The easiest BAD file only contains 3 columns: chromatin, start position, and position. There are also standard BAD6 BAD files with 6 columns, and also 12 columns. For max to output, the narrow peak format, it contains like 10 columns. WIG files and BAD graph files are coverage files. They show the number of reads fall into the region or the position of the genome. If you look at the content of the two formats, they are quite similar. Actually, BAD graph files can be converted into BigWig files, and they can be used for visualization. The name with the big in it are the compressed binary version of the original file. They are smaller and more portable. And hopefully, today's tutorial will help you better understand NGS data. Subscribe if you like it, and I will see you in the next video.